Hello again. It is Briandra with a little bit of post editing insight. Again, so during the transition to the second block of presenters, we again had some technical difficulties being able to switch the recording abilities of the live presentation. And so just really quickly, where it did cut off, I did want to say that we again were asking everyone to consider donating to our chapter of Synapse at the U of O. And we have a GoFundMe that we did start. And if you are willing to or are considering, you can just reach out to us. And also in the emails that we send out with the links to the PowerPoints and to the videos, we will also add in the GoFundMe URL for quicker access. Next, just before I passed off the hosting position to Lily Murch, that's when the difficulties started. So we weren't able to record her introduction for the next speaker. So I'm going to go ahead and do that for you now. So the speaker that you will see next is Jeremy F. Huckins, or Dr. Huckins. He is a PhD and is also a researcher and lecturer at the Department of Psychology and Brain Sciences at Dartmouth. Dr. Huckins will speak on his research and understanding of the default mode network. I hope everyone enjoys.
Well, you can take the same type of graph you can get from Facebook and apply that to the brain. So we have a whole bunch of brain regions. And then we can, sorry, just gonna try and, get the slides to the right spot. Okay, so if we take these different brain regions, we can go and map them out. So you can take all the brain regions and plot them down next to each other. So if each of these brain regions, the correlation between them, we map as a spring. So each dot on the left here becomes a dot on the right. And then how close they are is how similar their time series are, or how correlated their time series are. And then we can map them by either where they are in the brain or what types of networks those brain regions are in. So here we would see frontal parietal network, visual, or default. So you have all of these different brain networks, and you can see how connected they are while they're at rest doing nothing else. So now, one thing that I do want to note here, since we're talking about the default network a lot, is that the default network is nicely coupled to both the frontal parietal and dorsal attention networks. And these are both networks involved in cognitive control. And one thing that we see is that at rest, or when you're not doing an externally directed task, the default network is more active. On the other hand, when you're doing an externally directed task, the frontal parietal and dorsal attention networks are more active, and the default network goes down. So these two networks kind of seesaw together, but even though they seesaw together, we are seeing that there's a lot of crosstalk between them, which is probably them synchronizing about when to switch from the default network being on to the frontal parietal network being on.
Can I ask a question just for clarity?
Yeah.
So the dots and the lines are representing the activity and the, what exactly is it representing?
Sorry. So each dot on the graph on the right is a dot from the brain on the left. So it's a brain region.
Right.
And then the connections between them are how correlated these different brain regions are over time while you're at rest.
The
So if they're going up and down together, they would be a lot closer. So like these frontal parietal regions that are really close would have their time over time, their activity would be going up and down in synchrony. Whereas these regions that are really far over here might be going up and down at different times from these regions further away from them.
Thank you. Yeah, that makes sense. And yeah, so that crosstalk, I kind of got into a little bit in the end of my talk. Well, sorry, no further questions.
No, you can ask more questions.
I'm more still thinking about it, like, because I got a little bit into the saliency network as a mediator between the two, but what do you think is causing that crosstalk, or is it just the two regions interacting without a mediator? There's a known.
So yeah, so I mean, I think the salience network does come online, and the salience is almost more of a persistence network for a lot of it. So the frontal parietal network comes on when you're trying to start a new task. So if you're like, okay, I really need to concentrate and study, and I'm going to look at this page, the frontal parietal network might come on. And then the cingulopercular network or salience network, same thing, would be on for the duration of time that you're trying to like look at that page. But then if you start daydreaming or something else, those two networks would kind of disengage and the default network would be turned back on. Does that make a little bit more sense?
Yeah, it does. And yes, thank you.
Yeah, of course. So one thing that I wanted to kind of talk about as far as the relationship between when you're doing stuff versus when you're not. So on the top, we have a lot of brain regions and how often they're co-activated. So brain regions are co-activated together are in the different colors. So all the green brain regions are co-activated together a lot during, this is across thousands of fMRI studies. Same thing with the orange. So if you have the, all the orange brain regions are co-activated together across studies. And this is, you're looking at task versus baseline. Now, on the other hand, if you look at brain connectivity, so how tightly coupled they are at rest, not doing any task at all, you see a very similar pattern and very similar grouping. So one of the big takeaways that I'd like people to have from this is that when the brain is at rest, your brain regions are still talking to each other, and how much they talk to each other is very related to how active they are when you're doing tasks that are externally directed.
So in that sense, is it exercised if we continue to use it, or is it just sort of like a baseline of what is happening in us to cause what we're trying to do? Like Ben asked if you can increase the communication between the default and executive, or so
yeah, so there's definitely a lot of work on that, and people have been actually, I think a good example of it is like learning musical instruments. You end up getting stronger connectivity between motor areas related to those musical instruments. Mm-hmm. So as far as changing connectivity between specific networks, you would probably want to do tasks that activate both of those networks. So like some sort of specific training where you're co-activating those two networks together.
Do you have any examples of what a co-activator task might be?
Well, say you're trying to do, I don't know, maybe some really tricky motor task that involves a lot of fine-grained control, along with something, if you see something on the screen, you need to stop doing it. So that would activate the frontal parietal network. So then doing some sort of task like that, you might end up being able to inhibit, you may be able to co-activate those two networks and then strengthen the crosstalk between them, because you need crosstalk between both networks to have good task performance for that.
That makes sense. You're activating the inhibition by focusing on something else and then shifting your focus to the task at hand. And yeah, that makes sense.
Great. So the next thing I wanted to talk about, and this is just really brief. So, you know, you can think of the brain like a social network. You have all these different groups, right? Well, people tend to be relatively, you know, clicky. They either hang out with work friends or high school friends, or, you know, they end up in these social circles. Well, you know, I might be dating myself here with this movie, but so if you look at how clicky the networks are, so it could be termed the Mean Girls effect. So basically, as these networks are more clicky, those individual networks function better. If there's too much crosstalk between these networks, they actually don't function as well. So, you know, unlike in social circles where we would like people to maybe be less clicky, in the brain, you actually really want there to be a lot of distinct divisions between which regions talk to each other. And that's actually something that's been observed in a lot of different cognitive deficits, where you end up with reduced modularity. So like after stroke and things like that, you end up with brain regions being more liberal with what other regions are talking to. So basically, we want the brain to be like Mean Girls. So next, I don't know about anyone else, but anyone else get hangry? Because I definitely do. So if I don't eat for too long, I get pretty hangry. And also, you know, I have my coffee right here. Coffee's kind of a little bit of my lifeblood. But a really interesting question is, does food and coffee actually change how your brain is connected at a given moment? So to look at this, Russ Poldrack, who's a researcher at Stanford, went in and every single Tuesday for a year and every single Thursday for a year, and this is Russ, he put himself in an MRI, which I gotta say takes a lot of dedication. So a little crazy, very impressive. I think it was like 2-hour scan sessions. So, you know, he a couple hundred hours across the little bit over a year that he did this. But on Tuesdays, he didn't eat food before he was scanned, and he didn't have any coffee. Where on Thursdays, he ate food and he had his coffee. So what I'm going to show you next is the brain network differences within the same person just between the Tuesdays and Thursdays. So what we can see here is on Tuesdays, some of the networks are pretty similar, right? So this one's pretty similar, yellow one looks kind of similar, but we get a very different grouping in this red network here. So this red network is actually somatomotor. So it seems that when Russ was fed and had his daily coffee, his somatomotor network was actually less synchronized. And then also the connectivity between networks was different between eating food and caffeine.
So how does that translate to what we experience, if that makes sense? Like less synchrony in those networks does it just mean, well, what does it mean?
Well, so that's a bit of an open question.
Yeah.
I believe in self-report, Russ said that he drinks coffee every day so that he did not feel great when he wasn't drinking coffee. So perhaps having the somatomotor network that synchronized, if I don't drink coffee anyways, I'm a little bit of a zombie walking around, not very coordinated. So, perhaps it's something along those lines of that things just aren't firing quite as they normally are and not as active.
Plus, there's the mood. I'm in a horrible mood before I drink my coffee. I don't know how that. Well, maybe they didn't look at that specifically in this set of circuits, but yeah, that.
In this particular paper, he didn't look at that, but I think he did take diaries every day of how he was feeling, and actually he also did all sorts of protein expression sampling and things like that. And I think they're trying to make the data set public. So there's a lot of really cool data for people to look at from that. So all of this work, just like looking at brain connectivity and what factors might impact it, this brings us to a little bit of where does resting state come from? So we know some of it is genetic. So your brain connectivity is more likely to be like your parents than it is other people's. Experiences across the lifetime impact it. So brain connectivity changes quite a bit as you're developing from, an infant all the way through your mid to late 20s. And then also your recent and current environment. So, if you're really stressed, as we talked about in the previous slide, if you haven't eaten and haven't had coffee. And we do know that resting state is stable from scan to scan, more or less. Month to month, it's pretty stable. If you're daydreaming during a task while you're doing something, your brain connectivity actually looks like resting state and some sleep stages. But now one thing that I kind of want to mention is that there are these impacts of, mood. If someone's in a coma, the resting state connectivity looks very, very different. If someone's moving in the MRI, it's like trying to take a picture. If someone's moving around, it doesn't work very well. Their physiological state. And then there are also a lot of individual differences in resting state, but these individual differences are more like on the order of a couple percent, whereas the similarity across people is much bigger than there are individual differences.
So in my talk earlier, I postulated, I think that I may have injured the network's underlying resting state. Has that been documented, such as with brain injury or stroke or aneurysm, perhaps?
Yeah, so that there's a decent amount of stuff that's more recent coming out of a lot of the VA centers around, looking at control veterans and ones that went through TBIs in combat. And it's nice to have that control group at least, because then you have people that have been through the same stressful experiences that could impact the connectivity. So there's been some work with that, and I believe connectivity between salience and default network was found, and one group found that. A few other groups have found that connectivity within the default network is not as synchronized. So normally the default network's acting more or less as one unit with some pieces kind of going off on their own, but in larger bits, people that had TBI, that say different parts of the default network would be going different directions at the same time. So they weren't as synchronized.
Yeah, that lack of synchrony, I guess, would be, well, I don't know. I just know what I had experienced, and it was a feeling of what was in my mind was not me. So, but again, I was maybe postulating.
Well, no, I think you're really onto something there, because based on the functions or the functions that the default network underlies, right? So thinking about yourself and memory consolidation and things like that, a lot of those you do see in TBI and things like that. So that would be something to, I know there are a couple papers that came out in the last 2 years that are pretty good on this that I can send along. And there are a few more that I think are under review right now from people I know. So.
Cool. Yeah. And I think I haven't heard it discussed as much. I believe other people may have experienced that, but like any other network, it can be injured or changed or made less synchronous, especially if parts of it literally are damaged. So
definitely. There was a really good paper by Josh Siegel about stroke and looking at recovery from stroke. And one of the things that they showed is the after stroke, the brain becomes less modular. So it's not, not the Mean Girls effect, but then as people recovered, the modularity of the brain tracked their recovery in multiple cognitive domains. And just wanted to talk about a couple of applications of resting state. So there you can look at individual differences. You can compare patient control populations. You can observe resting state connectivity across a whole bunch of different animals, ranging from mice all the way up to humans. And it's nice to be able to identify how kind of internal and external components of neurobiology interact. And this brings me to, does anyone have questions?
is the resting state, so I guess you answered it in the earlier slides where it's many states, or there's many paradigms which will elicit its activity, but is it something that's studied clinically or looked at clinically from your?
Yeah, and I just wanted to dissociate something really quickly there. So the default network is a network that's more active while you're at rest, but
Right.
Resting state connectivity, you can look at any network.
Right. Okay.
But yeah, people are looking at it in clinically quite a bit. It's one of the easiest things to do because you don't really need much of a task. You just put a plus sign on the screen, like I have here, and run the scanner for however long you want. So then you can compare groups very easily. Oh yeah.
I had a question about the fed state and the fasting state, and you mentioned, I could have this all backwards, but I'm not clear on why more synchronicity, why it was less synchronous with being fed and feeling better, and more synchronous by not being fed and feeling like crap.
That is a really good point. So if you're thinking about activity in the brain, so say you have, well, let's go with 8 fingers here. So you have 8 neurons, right? So if you have 8 neurons, they're all going up and down at exactly the same time together, you can't get much distinct information out of those 8 neurons that are all doing the same exact thing. Whereas if some are going up and down at different rates, there's a lot more information content in there. So having the red network here, the somatosensory network, being more distributed, so not being as correlated, that probably means there's more information content in there, so that the having food and caffeine probably reflects a more awake and kind of nimble state.
So, Dr. Huckins, following up on my earlier question about communication between the different modes, I'm confused how that works in relation to the Mean Girls effect, which would seem to suggest that communication is not necessarily a good thing.
So So it's a tricky line. So you want some regions to be highly correlated so that they can communicate nicely, but then you want other regions to be less correlated. So you basically want it to sort of match a social network where you have a bunch of people that communicate a lot over here and a bunch of people that communicate a lot over here. And then there might be a couple people that actually talk to both groups.
Okay.
So, and then in that case, you say you have the default network here and the frontal parietal network here. People in the default network could be talking, and then the people over here could be talking. But what you would want is you would want the people in between telling them what group should take over for the moment.
Okay. So it's like a middleman kind of.
Exactly. So I didn't get too much into it, but the term for that is hubs. So, just like if you try and go fly somewhere, you don't fly directly there. You're probably going to go to Newark or Chicago or Atlanta or another major airline hub before you go somewhere else.
Mm-hmm. Okay.
I was wondering if your answer was going to be that with the what we call mean girl effect, it was one of being more alert or hyper vigilant in terms of just trying to survive, like maybe having coffee and being fed or not.
Oh, you mean the network being more tightly coupled, being kind of like hypervigilant for that? Yeah, no, that definitely would be a possibility. I do think in this case it probably is that these brain regions are differentiating their activity more. These brain regions are across the whole motor cortex, so that probably means that you're able to better independently move, say, your hands and your feet and things like that once you've had your coffee.
And why are your pink regions, I forgot what they're called, on the opposite side in Tuesday versus Thursday?
So that, that was a display choice that the authors of that paper did. So I was not an author on this paper, but they, I think they did it to keep the blue regions in the bottom left and then the purple and yellow ones in the same spot. But just the purple and red kind of flipped.
So I just want to keep following up on this, on the hubs piece. Then is that what we're talking about as far as like learning a musical instrument or going back and forth between the two modes that strengthens the hub?
So it might not necessarily strengthen a hub specifically, but information would be flowing through them. And the ability of the hub to talk to both networks at the same time, or even, it might not even be just one connection. It might be multiple sets of neurons or brain regions that you have to go through to get those brain regions synchronized. Mm-hmm. So, yeah, I think as far as hubs go, where they are in the brain and also what brain regions they're talking to currently really impacts their activity and what brain networks they can control. And to a certain extent, that can be trained though.
Thank you.
Fantastic. Yeah. And so I hate to cut the discussion a little short. Jeremy, I wanted to touch base with you. Would you be willing to share your slides from your talk today with the group?
Of course.
So we can go ahead and distribute that and kind of keep up on that discussion.
Yes, of course.